Hello world. So this is my enhanced version of Neurocave. I enhanced it to better interactively visualize and explore neurogenomics data sets, such as this this one from the Allen Brain Institute. And so they have some donors that donated their brains to science, and they did MRIs on them, and they did various different types of MRI processing, including diffusion tensor imaging, as well as T1 and T2 weighting. And you can browse their MRIs right there. And you can also go browse the genetic gene expression analysis right here. So let's say my favorite gene is WWOX. I can search for it, and I can see which brain regions express it in what way. And so we have some green areas, some red areas there. You can download an app, the Brain Explorer app, that gives you a better understanding there. I'm working on this Neurocave app that kind of has a new, unique way of interactively browsing neurogenomics data sets like these. So you can control the sizes of these glyphs. You can unlink them, and I can have the brain. What I have here is I have the brain's anatomical coordinates marked out over there, and these are 950 regions from which they sampled tissue and ran it through a full genome microarray. And in the offset over here in the distance are the actual, is a t-SNE coordinate representation of the genes and the gene expression levels in the microarray. And in the right pane, I can do a close-up of that one, so I can put this information in the foreground. Actually, I want to do the UMAP representation. It's got some better separation. So, and you see that's colorized with a clustering algorithm. But instead of the clustering algorithm, if I'm interested in long non-coding RNA, I can select that. So, and now I can I have the protein coding RNA mRNAs in blue, and in this pink color I have the long non-coding RNAs. And I can make the coding RNAs translucent and dim, to really bring into focus the long non-coding RNAs, or I can make the coding RNAs go away entirely. And I can I can bring up the search panel, and I can do a search for WWOX over here, and there it is. So I can now click on it. I adjust the threshold for the edge threshold. So I now have two threshold sliders, one for intra-group, so this shows you the connections to other genes, and I can include the coding genes and make them translucent right there. And I can also set the intra-group threshold, so I can show the connections to the brain over there as in the part of in the close-up. So see, I can turn on animations and make the opacity of the edges reduced so I can kind of better see through the clutter. You can see which brain regions have a relatively high expression level of WWOX. See the superior frontal gyrus there. Click on it. Adjust the threshold. And change the threshold mode. So now it's displaying 5, 10, 16 neighbors. And now we don't want to see the intergroup neighbors, just the intragroup neighbors. There, so you can see the cingulum bundle expresses a good deal of WWOX. And click it, you can see its neighbors. These are the brain regions that are highly that have a high degree of connection, neuronally speaking. A lot of fibers connect these, so you can really see. You can focus on the gene the gene expression, intergroup edges, or you can take those away and explore the connectome or gene co-expression neighbors and of the regions or cells that are in the focus.